Hello students, very good evening. I hope you're all doing fine, doing great at your side. Today is 21st of July, Wednesday. As per our series, we are continued with our various topics within the quality assurance. As we can see that these subjects, this QA, drug regulatory affairs, all these subjects, they are quite subjective actually. There is much, um, we are not there are not so many techniques that we can learn. It's more about the dossiers, how the paperwork goes on, what are the various quality assurance in various other countries, how things are working out in them, how the maintenance is done, how the premises are done. So we can see in the previous lectures that we had been through, it was more about the technical side of it, that how we have to maintain the field of pharmaceutical quality assurance like GMP, QA, QC, quality management, material management, right? Uh, the premises management. We have very brief about the introduction also. Slowly, we will also touch various parts of regulatory affairs, that how drug regulatory affairs like FDA, their rules that are meant in USA and India, how they are different and how they could be beneficial for all of us, right? So let's start with today's lecture. That is manufacturing operation and control. So that was one of the objectives that we have to deal with it. So actually in the previous lectures, if we see, did I share this book? Yeah, I shared that book. So we are done with the A part, B part, in the premises, equipments we are done, material management we did also, quality management we are also done. So today we are doing the F part, that is manufacturing operation and control. So almost we will be done with the 40% of your course by this, and we slowly discuss about documentation, pharmaceutical validation and so on. Uh, So as we know, the GMP which we are currently acceptable should be followed during the carrying out all manufacturing operations and the control. So there are four key factors in them in the GMPs. That is purity, identity, strength and safety. So on basis of four these things your GMP is based on. So during that GMP, your sanitation of your manufacturing premises is the main thing. So initially, so all these processes are interdependent with each other. Your industry or premises have to be clean, keep it clean, and there should be some standard operating procedure that how and when which chemical should be used, for how long we have to make it, and yeah. So for sanitation of manufacturing premises, that is the SOPs for that, standard operating procedures. So to define detergent and its concentration. So in our home, when we are cleaning or in any other industry, local industry, they just put pocha and they put, what we call, they add phenyl and it goes straightforwardly for cleaning. But in a higher MNCs, you have to maintain, proper level of detergent and its concentration, and the frequency of cleaning has to be taken care of, for how long in a day, We have to clean it 3 times. If we are doing it, at what time has to be done, what should be the method of cleaning, and the person who is responsible for doing it, supervising the job. So all such activities must be supervised by the housekeeping supervisors. Housekeeping supervisors, they are being taken care of all the sanitation of manufacturing premises. So sanitation of manufacturing premises, so like you have various different bags for different there's a bag for recycling, there's a bag for organic compounds or biological compounds, and there's a bag for plastic. So all these bags has to be separated from each other and then sent to the outside truck. So there could be some mix up and source of contaminations while in your manufacturing process. So in pharmaceutical manufacturing, mix up and contamination, it's one of the major danger. So proper precaution measure has to be taken care to avoid that the same. Right. Mix up can be defined as the presence of undesired materials into desirable materials, which can be generally be visibly seen. Example: cartons of one product into another. This could be also one thing could possible, and tablets of one product with another product which have similar size, shape, or color. So, what are the causes that may give rise to this mix up? So, the close proximity, they are quite near to each other, might be having same color, multiple products handled at same time, use of same apparatus or equipment for multiple products, improper storage of sterile and non-sterile products, care not being taken care during the packaging or repackaging of products, mistake made when dispensing them, and unused labels or earlier production left in the production area, improper documentation and labeling, or no ID on materials or products are given there. So causes that may give rise to mix up. So inadequate training of personnel, failure of the processing equipment, uh, manual packing and repacking, inadequate control procedures, improper affixing of labels, yeah, improper or lack of proper communications, personal handling of two machines at the same time. So all these things are one of the reasons for the mix up. So some source of contaminations, what could be? So contamination is also presence of some material where it is not desired, but it may or may not be visibly seen with the eyes. So like fine dust of one product going into another, fine black particles of dust into the processing materials, etc. So cross contaminations, contamination of starting material, intermediate products or finished products with another starting materials or a product during the production. So contamination of products can be caused by contaminated clothing, contaminated equipment, contaminated faculty HVAC system, so people carrying harmful bacteria, contaminated premises, processing operations, and utilities and services. Controlling of contamination and mix up, so exhaust system with proper air filtration and dust collection should be placed where heavy dust is generated. Collected dust should be disposed of by suitable method. Separate air handling unit should be provided for each workstation's activities so that a closed circuit air system along with a proper air filtration will be operating. This will avoid particulate contamination from one processing area to another. So depending upon the nature of the products being handled, different rooms should be maintained at different air pressures. Generally, more clean air should be maintained at higher air pressure so that the less clean air will not enter the cleaner air area. So doors of the room should open in high pressure area so that they always remain closed. So this is an addition to the provision of the door closures. So packaging lines in the central packaging area should be well segregated. A partition of at least 1.2 to 1.5 meters is recommended in two adjacent packaging lines. So these avoid accidental mix-ups of materials from two adjacent lines. So trained people in pharma processing the people should be trained in their jobs and also in the principle of CGMP. So following the correct and disciplined procedures and good manufacturing practices helps in avoiding the cross contaminations and mix-up then. Providing appropriate air locks, pressure differential and air extraction, so minimizing the risk of contamination caused by the recirculation or re-entry of untreated or insufficiently treated air. Wearing protective clothing in areas where products with special risk of cross contaminations are processed. Using cleaning and decontamination procedures of known effectiveness as effective cleaning of equipments. Using a closed system for material handling and productions, and so on. So what are the processing and intermediates bulk products has to be taken care. So starting from the receipt of the raw materials till these materials are converted into bulk goods ready for packaging into the primary and then finished packs, certain points are required to kept in mind so that the identity, strength, safety, purity of product is maintained. So following are some of these points. So like before starting, any processing and materials received from the store should be checked for identity and quantity. This verification can be done against the labels on their containers and by actually weighing or measuring the quantity of materials. So process area equipment must be clean and no traces of previous product should be there. Environmental conditions must meet the processing requirements. For example, temperature, relative humidity, pressure gradient, class of air and so on. So all primary containers used for filling the finished products should be cleaned to acceptable level of cleanliness. Bulk containers should used for storage of in-process materials should be also thoroughly cleaned and before use. So what are the processing and intermediate bulk products? How we do that? So all measuring, weighing, recording and controlling the equipments and instruments should be calibrated regularly so that always remain in the state of calibrations. Record of such calibration should be maintained. Repairs and maintenance operations should not present any hazard to the quality of the products. All IPQC checks should be carried out, recorded, investigated. So access to production areas should be restricted only to the authorized persons. So IPQC in manufacturing and packaging. So standard operating procedures should be available and followed for in process quality control activity during manufacturing and packaging activities. So critical steps should be identified in each formulations, processes and specifications defined for the parameters to be tested at each stages. So quality control equipments looks like this: fibrillator, dissolution test apparatus, digital pH meter, moisture analyzer, disintegration test apparatus, hardness tester. So all these six different have the different applications in the quality control equipments. So IPQC test for various dosage forms. So how you test that how much dosage is required according to IPQC. So drug content determination, then you moisture content of granules, then assay of the active ingredient is it present, then you weight variation of uncoated tablets, then hardness test and disintegration test. So all these 6 parameters make the tablet at the end. For syrup and suspensions, it's a different. For them, what you have to do is drug content determination initially, then assay of active ingredients, then pH, then weight per ml, then particle size. So all these 5 things has to be matters a lot. So IPQC test for various dosage form, that is semi-solid. So drug content determinations, assay of active ingredients, uniformity and homogeneity test, and viscosity and specific gravity tests. So all these matters a lot. So IPQC test for various dosage forms. So drug content determination, clarity test, pH, pyrogen test and leakage test. So packaging operation includes similar to the Manufacturing operations, packaging operations should also have to follow certain precautions. So avoid the risk of cross contaminations and mix up. And packaging equipment and line must be cleaned before starting a fresh packaging activity. Line clearance SOP should be followed and recorded, maintained. Each packaging equipment and line should be identified by a conspicuously fixed board and indicating minimum following information like name of a product, batch numbers, pack size, and date of a packing. Printing on labels, cartons, coated tablets should be carefully planned and activities clearly segregated to avoid mix-ups. So online verification of all labels by automated electronic beam can be helpful in preventing the mix-up. An empty packet detection system and tab should be used also. So printed and embossed information on packaging material should be distinct and resistance to fading or erasing. Online packaging check should be carried out at least for the following things: general appearance of the package, completeness of the package, correctness of the pack and packaging material, correctness of the overprinted details, correct version of printed packaging material is being used, and weight check of carton and chipper may be useful. So products which are involved in unusual event during the packaging operations should be fully investigated, record made and only when the authorized person is fully satisfied. So any significant or unusual discrepancy observed during the reconciliation of the quantity of bulk products and printed packaging, number of units should be investigated, and upon completion of a packaging operation, any unused batch coated packaging material should be destroyed and destruction recorded. So documented procedure should be followed if uncoated printed materials are returned to stores. So release of finished products. So releasing of finished product is the last activity in the manufacturing and packaging operation at the factory. So once the finished product is released, then it can move into the commerce freely. Hence, this is very important activity and should be carried out with the most care. Then your release of your finished products, while doing these points should be considered when you have to release your final product. So finished product must be placed in quarantine. Sample of products should be taken at intervals and must be retained for examinations by the quality control laboratory. Documentation has to be done, all the QC has to be taken care, and the QA department has to take care that the finished product should be released by sales by the authorized person. So some SOPs while releasing the finished products. So we can see that each step so far we have studied here is very consistent, is very deep, and it has to be channelized with the proper way. Proper documentation is always there, proper protocols has to be maintained, and a proper level of discipline has to be there also. So all these, if you want to work in future in the pharmaceutical line, in the pharmaceutical world, so be ready with that. Be a disciplined person, be a accurate person, be a concise person, and do your job honestly, of course. And anything that you find that something is misleading, mishappening, take your action immediately. Make your superiors to know that this thing is not the way as regular it was happening, because this will really help many company people also. If anything goes wrong into the market, you can save many life also. By mistake it could happen, some adulterant could be get into your drugs. So these things, so we are just mainly understanding the various paperwork that are going through while making your manufacturing and quality control at that. Yeah. So who is the authority to release this batch? He may be authorized person as per regulatory requirement, QA head, any other person suitable authorized for this person. So before releasing the finished product, at least following points has to be taken care: a complete BPCR, IPQC report, test or report of analysis, deviation report if it is there, and sterility reports. So process deviation can be defined as a variation in the production or any other process from the predefined procedures. So that we are thought that we will have that 200 milligram of tocopherol and 300 microgram of let's say any other ibuprofen in it, but it somehow the ingredients got changed, there is a deviation. So such deviation will have adverse effect on the quality of a product, and these should be avoided. And exceptionally, if required, then they should be justified and properly authorized and recorded. That if we have made like some change in the content of a tablet, then in the market it should be available in that way. So if a particular type of deviation is occurring frequently, then such deviation must be fully investigated, and if required, this may be permanently changed through the change control of procedures. Right. So the main purpose of written manufacturing procedure is to build the desired quality into the products. That means to assure the drug products have identity, strength, safety, purity, and represent to have any deviations from these procedures. An identified control process should be followed and documented, and any deviation from this will be justified, corrected, and steps taken to avoid changes. So there are types of various process deviations. So first is your planned deviation, then there's unplanned deviation. Planned deviation is the one which are described in pre-approved deviations from the current operational documents. So covering a specific period of time or number of batches, planned deviation shall be approved before execution. Then comes your unplanned deviation. So unplanned deviation is also called as incident. It is defined as an unplanned or uncontrolled event in the form of non-compliance from the design systems or procedures at any stage of manufacturing. So packaging, testing, holding, and storage of drug products due to system failures. So why investigate deviations? To prevent reoccurrence. So failures are cost to company. Failures can have adverse health impact on the consumers if go undetected. To identify the cause and take corrective actions. To identify other similar situations, take preventive actions. Continuous improvement and regulatory requirement. So basic process flow of deviation handling: identification of deviation first, then immediate correction of the initial impact assessment, then define, classify the deviation, is it minor, major, or critical, then do the following work on that. And it comes to the next topic, that is drug product inspection. So an activity of measuring, examining, testing one or more characteristics of a product, comparing the results with specified requirements in order to establish conformity of each characteristics. So objectives are to enforce good manufacturing practices in the pharmaceutical manufacturing facilities, to authorize the manufacturing of specific pharmaceutical products, to monitor the quality of pharmaceutical products in distribution channels from point of manufacturing to the delivery of recipient. So the same rules that we were discussing yesterday, drug product inspection, that is a 21 CFR part 211. So in here we have package and label products shall be examined during finishing operations to provide assurance that containers and packages in the lots have the correct label. A representative sample of unit shall be collected at the completion of finishing the operations and shall be visually examined for the correct labeling. So results of these examinations shall be recorded in the batch production or control records. So 5 steps methods of inspection. First is a routine inspection, then is a concise inspection, then follow-up inspection, special inspection, and investigative inspection. So what is the frequency and duration of these inspections? For all companies, inspection should be carried out regularly, like once a year. For large companies, marketing wide range of product inspection may be spread into several visits over a longer period, and the length of inspection depends on the size of company and purpose of visits. It may also depend on number of inspectors, and it may extend from days to weeks. Then expiration dating. The responsibility of the pharmaceutical manufacturer is that the drug product should have the stated potency and therapeutic effectiveness till the end of the shelf life of the product, and this shelf life should be based on the stability of studies data of the product concerned. For this purpose, a detailed SOP on carrying out drug stability studies is required, and the data so obtained should be used to establish the shelf life of each product before it goes into the market. And the expiration dates shall be related to any storage conditions stated on the labeling as determined by the stability studies carried out. So factors affecting stability of drugs: moisture, hydrolysis and pH, oxygen and oxidation, light, temperature, and then microbes. So expiring your dates, if the drug product is reconstituted at the time of dispensing, its labeling shall bear expiration information from both the reconstituted and unconstituted drug products. For example, there is a dry syrup preparation. Some eye drops are also labeled as how long they can be used after the opening the bottle pack. This is an addition to the expiry product of the unopened containers. Both above or the final expiry date, whichever is applicable, should be printed on the label or carton. Any appropriate primary packaging material. So, because when the air goes into your chemical, into your drug, this might create a faster expiry to your product. Then calculation of yield. So actual yield and percentage of the theoretical yield shall be determined at the conclusion of each appropriate phase of manufacturing. So processing, packaging or holding of the drug product, such calculation shall be performed by one person independently verified by the second person. So to meet these requirements, each critical stage of the manufacturing process should be identified. Theoretical yield at the end of that stage should be established. The actual yield at the end of the stage should be calculated, recorded in BPCRs and compared any abnormal deviations. So for the common formulation, such critical stages are as follow. For capsule, packing, mixing and filling. For tablet, packing, granulation, compression, coating. And for the liquid orals, packing, filtration, filling. In this way, things follows. So calculation of yield, theoretical yield. So an amount of product calculated from the complete reaction of the limiting reagent is called theoretical yield. So actual yield, the amount of product obtained when the reactions takes place. And the percent yield, the ratio of actual yield to the theoretical yield. So percent yield, that is actual yield divided by theoretical yield multiplied by 100. And change control. Change control is the process of handling proposed alteration to items that have been previously designated as fixed, and the procedure that controls the change is called change control. So changes refers to any modification equipment, manufacturing materials. So according to WHO, change control is the formal system by which qualified representative of appropriate discipline reviews proposed or actual changes that might affect a validated status. So the intent is to determine the need for the action that would ensure the system is maintained in a validated state. So change control is a procedure that ensures changes are implemented in a controlled and coordinated manner. The change control programs evaluate all changes that could affect the production and control of the drug product, intermediate or API. So it is the most critical element in the overall quality management of the pharmaceutical industry. So change control system provides checks and balances in the quality system by tracking and reviewing and proving the changes in adequate change control procedures, which ends up in the regulatory non-compliance. So these are the functional groups that are involved in change control. I hope class is going fine. Just checking that the streaming is going fine. There is no problem in that. Okay, good. Quality assurance, quality control, laboratory stability unit, labeling control, packaging, medicinal clinical affairs, regulatory affairs, marketing and sales, manufacturing. So all these are the functional groups that come under the change control. So changes in the pharmaceutical operations includes change in location equipment, batch size, processes, control parameters, engineering, change in any part of equipment, design and layout facility, research and development, change in specification of raw materials, quantity of raw material, change in shelf life, environmental conditions, stability protocols, stability conditions, etc. Quality control, change in the method of analysis, change in sampling plan, change in hardware of computerized instruments. Quality assurance, change in validation protocol, SOPs, documents, and sampling, change in cleaning aids, agents, and procedures. So significance of change, the major includes influences product's quality or process reliability, and the minor includes influences are as a unit required control. There's possible measures, official license, new approval, revalidations, in the minor amendment review documentations. So not requiring controls, no relevance to GMP authorizations. Then change, control change procedure. So initially you have a closure, then need for the change, then plan for the change, then you control the change, then implement or reject solutions. So SOPs for change control program, first is the purpose, objective, scope and responsibility. So in the purpose, to provide instruction for change control procedures, in objectives to provide a documented procedures for change control procedures, and then in scope is applicable for change control procedures, and responsibility your primary officer, your supervisors of respective departments, so overall respective department heads. So change control forms, section A, B, C, and D. So return drug products. Return drug products shall be identified as such as held if the conditions under such return drug products have been held, stored, or shipped before or during their return, or if the condition of the drug product, its containers, cartons, or labeling, as a result of storage or shipping, cast doubts on the safety, identity, strength, quality, or purity of the drug product. So the returned drug product shall be destroyed unless examination, testing, or other investigations prove the drug product meets appropriate standards of safety, identity, strength, and quality or purity. A drug product may be reprocessed provided the subsequent drug products meets appropriate standards, specifications, characteristics. If the reason for the drug product being returned implicates associated batches, an appropriate investigation shall be conducted in accordance with the requirement procedure for the holding, testing, and reprocessing of returned product. Drug product shall be writing and shall be followed. Then comes your next step, that is your reprocessing. It is a treatment of batch or sub-batch materials which are unacceptable quality by repeating the same process steps from defined stage of production so that the quality may be acceptable. So we have to maintain the quality. Yeah? So the occasional repeating of one or more process steps during the manufacture after it was known that preset limits had not been met, or there was an unexpected process problem is an acceptable part of the process and a rational reaction to the results obtained. So some ICH Q7 guidelines, go through them. Then drug product salvaging. Drug product salvaging, it means that drug product that have been subjected to improper storage conditions including extreme temperature, humidity, smoke, fumes, pressures, age, radiations due to natural disasters, fires, accidents, or equipment failure shall not be salvaged and returned to the marketplace. So whenever there is a question whether drug products have been subjected to such conditions, so salvaging operations may be conducted. So evidence from laboratory tests and assays that drug products meet all applicable standards of identity, strength, quality, and purity. Evidence from inspections of the premises that drug products and their associated packaging were not subjected to improper storage conditions as a result of the disaster or accident. Right? So handling of the waste and scrap, subjective is to handling the production that assumes a great significance in the pharmaceutical industry, since the use of incorrect destruction procedures have serious adverse consequences for the manufacturing unit if such material finds its way in the wrong hands. Yeah, therefore, it is utmost importance that inherent dangers of mishandling of reject scrap is understood by all persons concerned with the production's operation. In the scope, this part provides an overview regarding the generation of reject, its collection and accounting, and recommends guidelines for dealing with the rejects and their disposal. So the responsibility may depend on the following people in the pharma industry: employees in the production unit, representative of QA, and housekeeping staff. So scrap like rejected foils, bottles, cans, tins, which could have a resale value. Scraps are generated at various stages during compression, encapsulation, coating, and packing stages. In process checks, rejected printing, packing material from floor sweeping, expired or damaged goods, excess sample in QC after the test, and product sample from R&D at development stage. So pharmaceutical waste is potentially generated through a wide variety of activities: healthcare facility, general compounding, partially used vials, syringes, and fourth preparation discontinued. Then trash, this should be discarded or disposed by suitable means and do not have a resale value, like dust, unsellable materials, outdated pharmaceuticals. So regulatory bodies that see everything, your all the waste management of a pharmaceutical thing: Environmental Protection Agent, Department of Transportation, Drug Enforcement Administrations, Occupational Safety and Health Administration, state environmental agencies, state pharmacy boards, and so on. So types of waste. So on the basis of states: solid, liquid, gaseous, right? So on the basis of effect: hazardous waste, biohazardous waste, and radioactive hazardous waste. So all these has to be taken care. So types of waste on the basis of state: solid, liquid, and gaseous. On the basis of effect: hazardous waste, biohazardous waste, and radioactive hazardous waste. So the solid waste that contains substantial potential threats to public health or environment must meet any of the following criteria, right? Specifically listed as a hazardous waste by EPA, exhibit one or more of the characteristics of hazardous waste, and is generated by treatment of hazardous waste. Storage requirement: containers must be in good conditions, container must be compatible with the waste, containers must be handled in manner to prevent leaks and pills, containers must be inspected, and containers must be labeled hazardous waste or list the content of the container. So characteristics of the hazardous waste: uh, there should be corrosivity, reactivity, toxicity, ignitability. So all these things are matter in the matter of hazardous waste. So biohazardous waste is also there. That's a solid waste that contain or may reasonably be expected to contain pathogens or sufficient um, virulence and quantify the exposure to the waste of the biosusceptible host and could result in an effective infectious diseases. So this waste includes such as materials as used sharps, body fluids or materials mixed with body fluids, bandages or other materials that have come in contact with the body fluids. So methods of product disposal: high temperature incarnation, right? Incineration, waste immobilization, that is encapsulation, uh, inertizations, discharge to sewer, landfill deposits, chemical decompositions, medium temperature incineration, and open container incineration. So product disposal: printed packaging disposal and general trash sewage. And sales of product disposal: it could be large scale destruction, medium scale destruction, and small scale destruction. So schedule for M guidelines, all biomedical waste shall be destroyed as per the provision of the biomedical waste rule 1996, and additionally precautions shall be taken for storage and disposal of the rejected drugs. So hazardous toxic substances and flammable materials shall be stored in suitable design and segregated in closed areas in conformity with the central and state legislation. So these some references. So save trees, save environment. So that's the end of it. So manufacturing operations and controls, right? So that's it for today, student. I hope um, yeah, so we see us then again tomorrow, same time, same place, and continue with another topic, no detailed manner, and so on. Okay, thank you then. Bye bye. Wish you all good night. Bye.